So I'm Kim Goodspeed. And I introduced myself earlier, but I'm a child neurologist here at UT Southwestern and working a lot in the gene therapy space with rare diseases, trying to figure out how do we design a clinical trial? And one of the very first steps is a natural history study. So I just wanted to take a little bit of time because I think words like this get thrown around a bit. And sometimes it helps to go back and figure out, why are we even talking about it? And why are we asking you to do all these extra things if you've already been to the doctor a million times? So I just want to start with rare disease. I'm probably preaching to the choir, and you guys already know all this because you're living it. But I think some of these numbers are striking. This comes from Global Genes. But rare diseases in and of themselves are rare in isolation, but if you put them all together, they're not actually that rare. So there's 350 million people that suffer from a rare disease globally. And 8 in 10 of these rare diseases are caused by a particularly faulty gene or a mutation or a genetic syndrome. And these disproportionately affect children. So 1 of 2 patients diagnosed with a rare disease is actually a child, a pediatric patient. And many of these disorders are neurodegenerative, which I think it's heartening to hear the data that's coming out of Dr. Porter's group, that y'all's disease does not seem to be neurodegenerative, but many are. And 3 in 10 kids with a rare disease don't live to see their 5th birthday, which is just tragic. And that's really what pulled at my heartstrings of wanting to work in this space and try to see what else I can do to help and kind of move things forward. So if you have a collective voice, you tend to be louder, right? So squeaky wheels get the grease. So what makes things a little bit messier in rare disease? Part of it is how the information gets disseminated to people like me and Dr. Liu and Dr. Porter, because a lot of us see interesting patients in our clinic and we're academics and we get excited about new findings, and so we'll write a case report. Case reports will stack up, but case reports are written from the perspective of whoever wrote them, right? So whatever I find interesting may not be the same as what Dr. Porter finds interesting, as what Dr. Liu finds interesting. And so we may leave out other details that are super important. That can lead us to not having good biomarkers. So we'll get into a little bit what a biomarker is, but essentially an objective way to measure a disease. The clinical care between even different states in the US can be quite variable. And certainly when you're looking at the globe, access to care, standards of care are completely different from country to country. Many of us are using off-label therapies because we don't have medicines that are particularly geared towards a specific disorder. So we're using our best guess based on other patients with similar problems. So we've got off-label therapy use, which can differ from site to site. And then finally, if we take a step back, we've got lots of different stakeholders. So I think most importantly is you guys. So I'm really excited that we still got to do this today, even though it's not in the same format that we thought it would be, but you guys are the most important people, right? Like, we do this for y'all. So y'all are stakeholders number 1. And Kim Nye has done an amazing job of organizing the group so that you have a place to come and put your voices together. But there's also us in academics who have our own career goals and needs and objectives. And then you bring in an industry partner, which is super important to push things all the way across the finish line, but they have their own goals and objectives. And so you've got lots of different people that are trying to mostly hit a collective goal, but there may be different ways of getting there depending on what viewpoint you're taking. So, it doesn't like my things. Well, there. So that gets us to thinking about the drug development pipeline. How do we get a new treatment for patients? I stole this concept from a set of FDA slides, which I thought was really good. So you want to always begin with the end in mind. So as we're thinking about a natural history study, which is one of the first steps, and we'll dive into that in details, why are we doing it? We're doing it so that we can have a new drug approved. How do we get a new drug approved? And that's kind of the framework that we're all trying to think about it from. What do we need to measure now in order to prove a drug works later? One of the first things they teach us in medicine is that you can't know abnormal until you know normal. And so the first 2 years of medical school really are spent learning the normal human body. And now we need to know what is normal for each rare disease in order to be able to demonstrate that a drug changes that normal for the disease. And so what the work that Dr. Porter's already started lays the groundwork for another step to really hone in on those things that are abnormal in y'all's disease. And how do we then decide this is our baseline and our treatment takes us here? And how do we prove that it did? So the more traditional way that drugs used to be developed, and still are, was more of a linear concept. So you started in the basic sciences. There may be some translational studies that were done. So that's work in like cell lines or in a basic scientist lab, or work with mice like what Dr. Bailey's doing, and really looking to translate that across to humans and say, well, what does this seizure look like in the mouse? And can we say that that's what the seizures look like in patients? Or autism variants in mice, and behavioral testing in mice? And what does that inform us about for humans? And then it would jump over this divide where then we would start the clinical studies. But what I think is a new way to think about things, and again, this is coming from the FDA, is really starting that clinical development in parallel with the drug development. And that's what will push us over to be a little more efficient with our time, so that while these important studies are going on in Rachel Bailey's lab, we've got your natural history study team that's trying to work out the details of what is our first clinical trial going to look like. And that's really where the natural history study comes in. And oops, oh, it lets me click. Okay, so natural history studies and clinical trial readiness are terms that I think are starting to be used semi-interchangeably. We're thinking more about it. Historically, I think natural history studies were thought of as, we just want to be epidemiologists. Think about like what Darwin did, and he was looking at all these different amazing birds and turtles and looking at what they looked like in nature. We're taking a little bit different of an approach now, and we're still using the term natural history study, but really the goal of that natural history study is that we want to develop the tools that we need to measure how well a patient is doing, so that we can then say, if we measure them without the drug, they look like this, and if we measure them with the drug, they look like that. And that's what we want. We want them to look better. So it's the tools that we use to prove to the FDA that our medicine's working. And so the main purposes I was thinking about as I was thinking about explaining this to you guys is we want to track what the disease looks like over time. So at different age points, and how does each individual patient look at different age points? And are there differences, like Dr. Porter is already realizing, the infants maybe have a very similar seizure pattern, whereas as they get older, that evolves a bit and we get more spread. What is that spread of the disease? So, what does the most severely affected patient look like? What does the moderate patient look like? What does a mildly affected patient look like? And everything in between. And then helping us plan for these future trials. That is really why we're doing all of this. So I think that it gets us a few different things. So one, we get a better understanding of the disorder. That may even give us clues into different things that might be going on with the citrate transporter in particular. It may give us improved clinical care that we can then share with all of your doctors locally and educate the communities at large of child neurologists of, hey, here's this interesting disorder. These are ways that we can help these patients better that might be different from your other epilepsy patients or your other developmentally disabled patients. We can measure, develop clinical outcome measures, which we'll talk about, in line with biomarkers. That might also give us new clues to develop other targeted drug development. Now, I have a gene therapy bias. I'm hoping that that works well for this disorder, but it may be that some patients need an additional something on top of gene therapy, or just like you guys are finding where certain combinations of medicines work really well for you. Maybe there's something there that we can learn that would be important, and somebody else has an idea of a new drug that they could develop to target that particular thing. And then ultimately pushing these new drugs into trial. So the whole point of this, which I think I'm beating this over the head probably, but we have to prove that the treatment works. And the feedback we've gotten consistently from the FDA is that, how do you know that that's not just what would have happened anyway if you hadn't given that medication? So how do we prove to them? We have to have adequate and well-designed research studies. So that is what that's the FDA language for a study that is good enough for them to view as controlled and reputable and could be reproducible. So they put out these bullet points, and I want to highlight just a couple that I think are pertinent to what we're talking about today. The rest get into a little bit of nitty-gritty from a science side, but I think things that are important to think about for you guys and why we're asking of you another natural history study or more clinic visits than you've already had. We need a valid comparison with a control group, and I'll go into what that means. And then we have to have well-defined and reliable methods of assessing a response. So those clinical outcome measures and biomarkers of how we measure and prove that a drug does something in a patient population. So what are control groups? So anytime you're doing an experiment, you want to know that I've got my experimental group that I'm doing some sort of procedure to, and then I have another control group that I'm not doing that procedure to, and I see what the procedure does because one group gets it and one group does not. So that's what we would call a parallel control group. So you might have heard of placebo-controlled trials. So that's an example of that. So we have a group of patients that come together, and then patients are randomly assigned to go to the treatment group or go to the placebo group. The treatment group gets the new experimental treatment. The placebo group either doesn't get a treatment, gets a fake treatment, or maybe just standard of care. There's a lot of different ways to do that. But the important thing is one group gets the experimental treatment, the other group does not. You watch them over time, and then you see what the groups look like compared to one another. Another way that we can do this in rare disease, because you don't have as many patients in rare disease. So for you guys, you've got about 100 patients in the world. And compared to something like diabetes that has, upwards of 1 million probably in the world. So one other thing that we can do is get a little creative with how we design our trials. So something called historical controls, which you'll see over here on the right-hand side of the screen. So this is where our natural history group would be super important and helpful to us, where they could potentially be the comparator group. And I use group somewhat in quotations. So the idea would be that you could follow patients for a period of time without a treatment, and then introduce that treatment to the same group of patients and follow them over time and see what changes. Do we see better adaptive behavior scores? Do we see improvements in their language skills, their communication skills? Are they walking better, running better, balance is better? And then we're comparing what that group of patients looks like to what they looked like before the treatment. But in order to do that and to prove to the FDA that that's real, that time period where we're following them before treatment has to be super standardized, not variable across institutions, not variable across countries. The patients have to be looked at in exactly the same way. And so that's why it's a little bit different than just going to your doctor visit, where they're tailoring the care completely to you and the measurements completely to you. We'd have to do the exact same measurements across each patient at each time point. And so that gets us to, well, what do those visits look like? And we can think about clinical trial endpoints, and I think of them in kind of two different boxes. So we have clinical endpoints and we've got surrogate endpoints. So the clinical endpoints are going to be ways to measure patients that, how they're functioning, how they're performing. And the surrogate endpoints, if you can imagine, in like a pregnancy surrogacy, somebody else is carrying the child for you. It's a measure that's indirectly measuring what we think is going on. So that may be blood levels of things, brain imaging findings, EEG findings, like what Dr. Porter just went through, where we can measure things objectively, but it's not directly telling us how a patient is doing. The clinical endpoints, on the other hand, are directly measuring how you guys are doing as a family, how the patient is doing from your point of view, how the patient's performing on different motor scales or tasks or language scales or tasks, things like that. And these two things can really be used in parallel. Sorry, let me go back if I can. Oop, nope. Okay, so they can be used in parallel. So we can look at what do EEGs look like in patients that have lots and lots of seizures versus patients that don't have as many seizures. We can look at blood levels and compare those to how patients are functioning. So they're really used hand in hand. Both are important. And these are the tools, just to say one more time, that the FDA will be looking to say, yeah, your drug is doing what you think it's doing and the patients look better. So the whole point, I think, of any kind of clinical research program is we want patients to be better. We want patients to feel better, to feel empowered, to have options. And so the way that we get to that better patient care is through all of these different pieces of a clinical research program. So the natural history studies, your patient registries, and clinical trials ultimately. Patient registries are, I stuck those in the middle of the diagram for a reason. I think they're really important to keep tabs on each other so that you know who's in your community exactly, what is your pool of resources, who are the patients that are interested in helping and participating and being involved. And those can be really important resources for designing natural history studies, designing clinical trials, and then also recruiting patients. So my favorite Dr. Seuss analogy here, I always think about this, Horton Hears a Who. So I think about rare disease as, this tiny little community on the tip of a thistle who individually, Horton couldn't hear them. But as soon as they started putting their voices together, he heard them, and initially people thought, his other elephant friends thought he was crazy, but eventually other people heard him too. And I think we're finally getting to this space where the world is hearing rare disease is important. And I will leave you with that. So I know that you guys are here and I'm excited to be part of it.
Thank you so much. That was so great.